Dr. Malene. It is a great pleasure to have Dr. Malene Lindholm here with us today to present on some exciting research. And it's my pleasure to give a brief background of Dr. Lindholm. Dr. Lindholm obtained her PhD at Karolinska Institute in Stockholm, Sweden in 2015 with Dr. Carl-Johan Sundberg, looking at exercise and epigenetics, and moved to Stanford University for a postdoctoral fellowship for 4 years in the group of Professor Yuan Ashley. And for 2 years was an instructor junior faculty of medicine at Stanford, and is currently director of the Wu Tsai Human Performance Alliance, Human Molecular Athlete Moonshot Program at Stanford, as well as a senior research engineer at Stanford as well. And as you're going to see today, is a leader in some very exciting research. And Malene, thank you so much for joining us today. And please feel free to share your screen, and yeah, we can go forward.
Yeah, of course. Thank you very much for that kind introduction, and thank you so much for inviting me to talk to you all today. So let's see if it works to
Our pleasure.
share the screen. Can you see that?
Perfect. Good to go.
Awesome. Yeah, so I'm going to talk today about the mitochondrial multi-omic response to exercise across tissue. And this being a muscle health group, I don't think I have to tell you how great it is to exercise. So what if we had a pill to do all of these different things, give us better cardiometabolic health, better sleep, better self-esteem, reduce risk for depression? And of course, we already have this pill. Exercise is really the ultimate poly pill. And we really want to try and get these all these different effects. But of course, we already know it's great for us to exercise, and hopefully everyone in this audience do. But how exercise exerts its many health benefits is not completely understood. And one of the reasons for this is that historically it's been pretty difficult to fund exercise-focused research, as most funding agencies have really focused on sort of the disease end of the spectrum rather than the health end. But fortunately, in the past 10, 15 years, this has been changing. Lots of big funding agencies and philanthropists are now realizing this gap and have started some big initiatives. And I'm going to talk about one of them today called MotorPack, for Molecular Transducers of Physical Activity Consortium. So this is an NIH Common Fund project that aims to identify the molecular drivers of the exercise response and looking at different levels of molecular regulation. But just some background, we started MotorPack about 8 years ago. And at that time, we didn't really know. We wanted to sort of establish what's the current status of the field from a molecular perspective. So we conducted first a meta-analysis to try and identify, okay, what are the omics data sets out there? What's the knowledge that we already have? And where are the important gaps? And what we realized primarily was that we only had enough data for transcriptomics from exercise studies to really do proper meta-analysis, and only from 2 different tissues, so muscle and blood. So we conducted this meta-analysis, and this just shows a screenshot from the associated web resource. So you can go in and you can search for whatever gene or you're interested in and see what happens across public data sets out there. What was nice with this was that we did have the power to start looking at things such as sex and age associated differences in response. We know that there is a large variability in responsiveness to exercise across many different parameters. And here, especially in the muscle, we could start to untangle some of the sort of sex and age associated differences, and also start to look some at these sort of time-wise trajectories of how different molecules change, or in this case, transcripts change in response to exercise. One of the findings you see there in the heat map is that we saw that with age, there was a greater inflammatory response so a greater inflammatory response to training in older individuals. So this indicated more of an enhanced cytokine signaling, but also increased antiviral activity. But what this really told us was that there is a lack of well-described data sets across primarily other tissues and also across other omes and transcriptomes. So this really highlighted the need for efforts like MotorPack. So I'm going to talk about this today. And there are two phases to MotorPack. One is sort of the preclinical or animal study part, where we have two age groups of rats. So we have 6-month-old rats and 18-month-old rats. And they have performed either a single bout of endurance exercise on a treadmill or training for 1 to 4 or 8 weeks. So we have a time series component to the endurance training. And then with lots of different samples taken from these rats. And today I'm going to focus mostly on the results we have from the 6-month-old rats, which were recently published. We also have a human cohort, and I'm going to mention some of the preliminary results we have from the humans. There is a large sedentary adult cohort, and these are randomized to either endurance or resistance type exercise. They then train for 12 weeks. They're either modality, and then there is also an untrained control group. So both before these 12 weeks of training and after, they do an acute bout with biospecimens collection. So from the humans, we take blood, muscle, and fat. And then after the training, they do another set, of course, of phenotypic assessments, and then another acute bout with biosampling. There's also a pediatric cohort as well as a highly active cohort as part of MotorPack. And we're up to 1,500 people in this sedentary adult cohort. But at this stage, we're just finalizing recruitment. So we're into the last 30 days of recruitment, something like that. So we're going to end up around between 1,550 and 1,600 people in the big cohort. And then we also have 200 additional people that were recruited prior to the COVID shutdown. So the human study was a bit delayed due to the COVID shutdown. So I'll show some preliminary data from those first 200 people today as well. But for the animal component, one of the key strengths of this is really the number of tissues that we have from these rats. So we've collected 19 different tissues in response to training. You can see the 19 different tissues here. It's a lot, and many of these have never really been looked at in response to exercise training before. We also looked across a lot of different levels of molecular regulation. So this is a multi-omic study. So we look at the epigenome using attack sequencing that assays sort of chromatin accessibility, reduced representation bisulfite sequencing for DNA methylation. You could look at transcriptomics using RNA-seq. Proteomics, we're looking for protein abundance using mass spec. And then we also have 3 different post-translational modifications and a targeted immunoassay. And a mix of untargeted and targeted metabolomics and lipidomics assays. So lots of different levels of regulation studied in these rats. So today, I'll talk a little bit of the findings with regards to sex differences. I'll, of course, focus on the mitochondria that I work mostly on. And then also mention a little bit on how we can use these multi-omic findings to try and identify potential therapeutic targets of interest, for example. So one of the most prominent things we observed when we started to analyze these rats and the training responses was that there were a lot of sex differences in virtually every tissue. In the end, we actually had to separate the analysis based on sex because the variability was so different between the sexes. But if we start looking at the phenotypic data, so this just shows the VO2 max response in these rats. Here I'm just showing the control, the 4-week and the 8-week animals, so not the 1 and 2 weeks, which is of course pretty similar to the controls from not that much happening at the VO2 max VO2 max response over just a couple of weeks. But what you can see here is that here is for males in blue, there is a robust increase in VO2 max as expected with endurance training, and same with the females. There was actually a decrease in the untrained control cohort. So these control rats remained sedentary for the 8 weeks that the trained group was exercising on the treadmills, and were exposed to the treadmills only with no exercise. You can see the body weight there, where the females increased in body weight, and then this is mainly driven by changes in body fat. So we see that the control animals increased in percent body fat in the females, whereas the trained animals retain their fat mass, as opposed to the male animals that lost fat mass with the training. And then you have the corresponding lean mass numbers there. So we know, of course, this happens. We've seen this a lot before, but we don't know exactly how. So for the sex differences, looking at all of the changes that we saw with training, almost 60% of the responding molecular analytes were sex specific. And we even saw opposite responses in some of these tissues. And in this figure, I'm just showing some of the immune-focused responses. So here you have immune cell enrichments. And here the direction of the triangle indicates the direction of change. So upward pointing positive enrichment and downward pointing negative enrichment. And the gray with the black, the gray triangle with the black frame is sex consistent changes. Blue is for male and pink is for female. So you can see a lot of tissues show divergent responses when it comes to sex. There was a particularly interesting response in the small intestine here. You can see that a downregulation of a lot of these immune pathways, and this was most prominent in the female animals. And we could also associate these with gut inflammation pathways, and to gain some insights into how exercise can prevent inflammatory bowel disease, for example. We also saw male-specific increases in white and brown adipose tissues. And when we looked at markers of these different immune cell types, we saw that this was associated with an increase in a lot of these immune cell types in male animals with training in both white adipose and brown adipose. So an infiltration of immune cells in response to training that was specific to the males. Another fascinating observation, also again coming into the mitochondria a bit, was the metabolic remodeling of the adrenal gland. And here we saw pretty much an opposite response between males and females. So this is the sort of largest time trajectory of change. You have the controls, and then you have 1 to 4 and 8 weeks of training for the females versus the males. And each line here corresponds to one transcript, because the adrenal gland is primarily assayed for transcriptomics. So we see this robust downregulation after 1 and 2 weeks in females, so quite early, and then it remains downregulated for the remaining 8 weeks of the training. The same transcripts in males increase after the first week, and then they go back to baseline. So if you just compare 0 to 8 weeks, nothing really happens. And we saw the adrenal gland was the top responding tissue out of all the tissues that we looked at, all these 19 tissues when you look at the 8-week training response, with almost 4,000 genes changing in expression. And these were primarily associated with mitochondrial remodeling, so energy metabolism as well as steroid hormone metabolism. And just to show really how striking this difference is, so here, this is the female, and you have the time points here from 1 to 4 and 8 weeks. And then the males, you can see this robust downregulation of all the mitochondrial pathways. This is a gene set enrichment analysis that's based only on the mitochondria pathways. So only mitochondrial pathways that now have been grouped into categories such as metabolism and small molecule transport. But you can see this early upregulation in males, and then it sort of subsides towards the end of the training period. And then you have this downregulation in the females that's really consistent across all time points. And if we look at sort of what genes that are affected, you can see this is the electron transport chain. We have beta oxidation and TCA cycle. And you can see pretty much all of these components are downregulated in the females, and either no change or slight upregulation in the male. So it's very, very different responses. And we try to also see what is driving these differences in the adrenal glands. So we did because it's transcriptomics based, we wanted to see what are sort of some of these upstream regulators that are controlling these differential transcripts. So you can see some of the transcription factors that were predicted to be responsible. So we have some of the estrogen-related receptors, the PPARs, which are, of course, known critical regulators of metabolism. And this was also super interesting in the context of how exercise affects stress response, because obviously we know that adrenal gland is releasing cortisol, and fit individuals don't get the same cortisol response to either exercise or other stress challenges like a mental stress challenge. So we also looked at the cortisol level, which in a rat is corticosterone. They don't have cortisol. And you can see in the males, the corticosterone really follows this changes in expression pattern with this early increase after 1 week, whereas in the females, it doesn't. And of course, cortisol will be more interesting to look at when we have in the humans, for example, where we have an acute bout in the untrained state, and then we have an acute bout in the trained state. Because here, I was actually not expecting us to see much at the cortisol or corticosterone level at all, because these samples were taken 48 hours after the last exercise bout. So we're not seeing any acute transient changes here. It's only sort of the baseline adaptation 48 hours after the last exercise bout. So it'll be really interesting to see what happens in the acute response in rats, and also in the humans, of course, once we have that, even though we don't have adrenal gland in the humans, but at least we have the plasma. So these results were published earlier this year in Nature and made the cover. So if you're interested in reading more, you can find it there. But given these large sex differences, primarily in the metabolic response, I wanted to further understand this from the mitochondrial perspective, and specifically look into mitochondrial adaptation. And of course, we know mitochondria, they're key organelles. They're involved in lots of different processes, not just energy production, but also apoptosis, calcium handling, ROS handling. There have been lots of amazing studies. I saw David Hood join the Zoom, done amazing studies on mitochondrial adaptation to exercise, and we know pretty well what happens in skeletal muscle, primarily, to some extent in the liver and the heart. And we see mitochondrial biogenesis. We see increases in mitochondrial quality with stimulating mitophagy, for example, improved metabolic capacity, ROS handling with the SOD2 induction. But a lot of these has been established from more targeted approaches, and we don't really know much about other tissues. So the goal here was really to look at this from the multi-omic perspective. So we have, again, these 19 different tissues, the multiple omes, and then we used mitochondria which is a resource that assigns basically either it can be mitochondrial transcripts, nuclear transcripts, and then associated proteins that are physically located to mitochondria. So it's not just the mitochondrial genome, but it also covers the nuclear genome with mitochondrial features that are physically located in mitochondria. And then we used a metabolite just a curation to associate, basically take the metabolites that are associated with mitochondrial processes, and did a targeted analysis of that. This was also published back in May in Cell Metabolism. So if we start by looking at these sort of multi-omic changes, so this heat map just shows the 19 different tissues here and the different omes that were analyzed. And so you can see here the transcriptome was analyzed for all of the 19 tissues, same for metabolome. And the numbers here correspond to the number of differential mitochondrial analytes in this particular tissue. So mitochondria includes almost 1,200 genes. So you can see for adrenal gland, it was about 50% that were differentially regulated. And just as the adrenal gland was the top responding tissue in looking at all analytes, it was also the top responding from the mitochondrial perspective. So we saw that there were some tissues that didn't respond very much. So for example, we have the 3 different brain regions. We looked at cortex, hippocampus, and hypothalamus. Didn't see much mitochondrial associated changes here. Of course, the brain gets a comparatively low increase in metabolic demand with training. It has high metabolic demand consistently compared to other tissues. Didn't see much change in the lungs, the ovaries, small intestine, spleen. But then there were some tissues that responded a lot more. So the adrenal gland we already talked about. Brown adipose tissue, colon was a really interesting tissue, was one of the top responding tissues from a mitochondrial perspective. You can see we have all assays in the heart, also in the liver, where we see a lot of changes across multiple omes, but primarily the protein and acetylome. So protein acetylation changed a lot. So acetylation of mitochondrial proteins. The skeletal muscle, we had 2 different skeletal muscle, the gastroc and the vastus, and then white adipose. So what are these changes? This is a pretty busy figure, so I will just talk you through some of the key sort of take-home points here. So this is a pathway enrichment analysis where we, again, have mitochondria pathways only. So all of them are mitochondrial pathways. We have the same as we had before for the immune regulation with a black framed arrow is for sex-consistent changes, a pink one for females, and a blue one for males. So what we saw was the expected upregulation of mitochondrial pathways in the heart, muscle, white adipose, and liver. Again, you have the tissues up here, so you can see the skeletal muscle and the white adipose here, and then you have the different omes down here for those particular tissues. In the brown adipose, again, we only had the transcriptome. We didn't have a proteome analysis for brown adipose, but there was a pretty striking downregulation. And this was more prominent in the male animal. So you can see both sex-consistent downregulation as well as male-specific downregulation of pretty much all mitochondrial pathways in the brown adipose. The adrenal gland, we already talked about this female-specific downregulation after 8 weeks. And here you can also see the changes in the colon, which were also predominantly in females, but here it's an upregulation of almost all mitochondrial pathways. And this was also associated with changes in lipid metabolism pathways. And you can see the lipid metabolism was actually one of the pathways that were affected across most tissues. So typically we think about the mitochondria as the sort of cellular engine, affected in working tissues, but this clearly shows that many mitochondrial pathways and processes are regulated across multiple tissues, and not just the classic working tissues. So I just wanted to highlight one particularly interesting tissue from a metabolic perspective was brown adipose. And here you can see the top 5 largest trajectories in brown adipose. So this is a way we use to try and visualize the main trajectories of the data across time. So here you have the 1, 2, 4, and 8 weeks of training. We basically binned or classified analytes as either upregulated in females only, upregulated in both males and females, in males only, no change, and then you have the downregulated side. So the larger these nodes, the more analytes belong to that node, and the same with these sort of edges between the nodes. So for the top 5 trajectories in brown adipose, we can see that the majority did not change for the first 4 weeks of training. And then there was this delayed downregulation between 4 and 8 weeks of training. And again, this is only for the mitochondrial analytes, only mitochondria. And most of them were in both males and females, but then there was some that were specific to males. We did see that this was associated with a downregulation of mitochondrial of markers of mitochondrial volume. So we looked at to try and assess mitochondrial volume more. We didn't have a direct measure of mitochondrial volume, so we used mitochondrial DNA quantification. We also looked at cardiolipin content for the where we had lipidomics data. And then we also looked at the percent reads from RNA-seq that came from the mitochondrial DNA. So that's just what I'm showing here for brown adipose. So you can see that there is a downregulation in both males and females, and this is more so in males after 8 weeks of training. So we were able to associate this late downregulation to a reduction in mitochondrial volume, similar to the female adrenal gland as well. We also looked at the thermogenic uncoupler, UCP1, and saw this was really striking opposite regulation in males and females. It's the light blue one here. You can see it follows this late downregulation pattern in the males, whereas in the females, there is a slight upregulation even of UCP1. And this coincides, both this delayed downregulation of all the mitochondrial analytes as well as UCP1 coincides with the main reduction in weight loss that we saw that was also specific to the male animals, or not weight loss, but reduction in body fat mass. So it could be due to some way for the male animals to preserve energy and prevent too much energy loss through thermogenesis with uncoupling. This is just another example, the liver, where again we have these across the 8 weeks of training and the top 5 trajectories, similar to what we saw for the males, or for the brown adipose. But here we have much more, many more ohms. So they're colored here by ohms. So you can see this large purple trajectory here that is upregulated in males only up to 4 weeks, and then upregulated in both males and females after 8 weeks. And this is the corresponds to the acetylation changes. So a lot of mitochondrial proteins increased in acetylation with training. And this happened despite a small increase in the main mitochondrial sirtuin, SIRT3. And we saw that this was specific to the acetylation level and not so much so this just shows the protein log fold changes. And if we want to see sort of which proteins that were actually affected, we did see this effect in a lot of the key bioenergetic processes in the liver. So here you have the beta oxidation pathway, the TCA cycle, branch chain amino acid metabolism, and then the electron transport chain. And this just shows the sex consistent changes, where each of these red lollipops correspond to an increase in acetylation of one site of this particular protein. And a blue one corresponds to a downregulation. So you can see most of the proteins increased in acetylation, and this was particularly prominent for the complex 5 proteins. And interestingly, this has also been seen in human skeletal muscle in response to high intensity interval training. So it seems to be something that occurs in other tissues as well. And we saw similar things in the heart, although not as prominent. And we do see, of course, a great increase in substrate flux through the liver, and also then the associated turnover of acetyl coenzyme A. And we did see this also to be associated with looking at the lipidomics data, with an enrichment in a lot of the lipid classes in the liver. So lots of data to untangle here, but this was something that was super interesting to us with these post-translational modifications that really haven't been studied that much in response to exercise before. So we continued this work and did a deep dive into the skeletal muscle data. So this is something that we're working on currently. So I'm working primarily with 2 very talented scientists from the Pacific Northwest National Laboratory, Tyler and Gina. And we have then now added, so initially we did not have acetylation data from the skeletal muscle. So we ran acetylation and then also redox proteomics from the skeletal muscle sample. And somewhat surprisingly, we didn't see much changes at all in the acetylation level. However, the protein oxidation did change a lot. And what was most striking was that there were a lot of sex differences in how the protein oxidation was regulated. So we just look at the proteomics first. We see these expected patterns. Here again, this is a gene set enrichment analysis. We see enrichment of mitochondrial proteins that are increased in both the females and the males with training. We knew this again from before. But if we look at the redox proteomics, we saw that an increase in the oxidation of mitochondrial proteins that was really specific to the male animals. So only the male animals increase in protein oxidation for mitochondrial proteins. And in the females, it was more glycolytic proteins that were oxidized. So this is something that we're currently working on, really trying to sort of entangle what does this really mean for protein function. This is something that we mostly don't know. So we're looking through both known literature and thinking about interesting follow-up experiments to try and identify how this is actually important for regulation of mitochondrial function in the skeletal muscle. So I hope I have convinced you that, even though the working muscles are very important, exercise really induces systems-wide effects, and how this happens really is still an open question. There is a lot of interorgan crosstalk, clearly. We've started to identify a lot of different exerkines that are secreted from different tissues and can have systems-wide effects. So this is something that we're also looking to identify through the human studies, and I'm pretty sure we'll learn a lot more about this in the coming years. So now I also wanted to spend some time on the human studies and primarily what we've done or what we've seen from a mitochondrial perspective. This is, again, pretty early data. We have, for the humans, this first batch of people, we're only looking at 1 bout of exercise. And the reason for that is that with the COVID shutdown, people had to stop training. So too few people were able to go through the entire 12 weeks of training. So here we have a resistance training group, an endurance training group, and then we have a sedentary control group. They have done one bout of exercise. We take the muscle, blood, and fat, as I mentioned before. So we take it before at baseline. Then there is an early group, a mid group, which takes like around 3.5, 4 hours after, and then a late that takes 24 hours after. So we have 3 different time points from the after exercise from the humans. In total, we're looking at around 200 people. So this is really, again, preliminary, but we're trying to identify these sort of main time-wise trajectories. So similar to the time-wise changes that I showed you in the rats, we're doing a clustering analysis to see what are the sort of key trajectories in the human data and what are the different ohms that are driving these. And so here we use all of the ohms for each analysis. So both proteomics, transcriptomics, metabolomics, etc. And there are lots of these different clusters with time-wise changes. This shows for adipose. So if we focus in on cluster 4, which is where the majority of the mitochondrial pathways are, you can see this is an enrichment analysis, again, pretty busy. But for cluster 4, you can see that this really peaks after 4 hours. So in adipose, the majority of the mitochondrial analytes increase after 4 hours, and then they're back to baseline and even slightly down after 24 hours. So here again, these are just mitochondrial pathways. So just looking at the mitochondrial adaptation, you can see some of the top most significant oxidative phosphorylation changes for the adipose. If we look at the same ones for muscle, as expected, we see this upregulation. Again, you have endurance and resistance. So we zoom in on the main clusters there, and you have sort of primarily cluster 9 and cluster 11 that show this delayed upregulation after 24 hours, and same here. So we're looking into what these do. Again, this is pretty preliminary. We have also identified some interesting exokines within these trajectories. So a lot more is coming from these studies in the near future. I also wanted to spend some time on how this is interesting and available to you. So one of the key, so I represent the Bioinformatics Center for MotorPack, and one of our key tasks within the consortium is really to disseminate all of this data and make it available for other researchers like yourselves. So we've spent a lot of time building what we call the MotorPack Data Hub, that with the purpose of making this data accessible and easy to interrogate for everyone. So I'll do a little bit of a demo here. You can see this is what it looks like. So you come to the MotorPack Data Hub. It's motorpack-data.org. And here you can do a lot of things, but you can download the data. So this is for the 6-month-old animals where the data has been published. It's available already. If you're interested in a particular ohm, like proteomics or transcriptomics, you can immediately download all of those data just like that. Say you really liked your experience. You can also, if you're interested in a particular tissue, like gastroc or heart, you can download all the multi-omic data from that particular tissue by just clicking there. So that's it's very easy if you want to play around with the data yourself. If you're interested in a particular gene, you can also, or several genes, you can go into the gene-centric view, and you can just search for whatever gene you're interested in, like succinate dehydrogenase. You can then select the tissues. So you just click on whatever tissue you're interested in and whatever ohms. And then you'll get that, and you can immediately visualize how what happens with the succinate in those two tissues. So you can see upregulation in both gastroc and the heart. Yeah, you can also search for multiple genes, as I mentioned. If you want to throw in some other tissue there, you can. And then again, you can visualize these results if you want to compare what happens. And now I selected cortex, so you can compare what happens in the brain, in the muscle, and in the heart. So you can see not much changing in the brain over these 8 weeks of training, whereas you see this robust upregulation in the gastroc, and a little slightly different pattern in the heart. So whatever you're interested in, you just go there and put in those that your favorite genes or proteins, metabolites. There's also differential abundance data, and just searching for the same gene here. So here, what's nice is you can easily also download the results that you're interested in. So for example, if you're interested in the multi-omic changes of succinate dehydrogenase, you get the time-wise changes. You can also look at training data, looking across the entire 8 weeks, and then you can immediately download these results. So if you want all of the multi-omics result only for succinate dehydrogenase, you can just download that immediately from the Data Hub. Some other cool tools, we have a graphical visualization tool. Here there is also a mitochondria specific one. So you can select whatever tissue you're interested in, like the liver. Hopefully you'll recognize these time-wise trajectory changes that I showed before. Here it is with that's colored by ohm. And then you can see for this sort of top changing trajectory in the liver, you can look at what are the top pathway enrichments per ohm. So you see for proteomics as well as acetylomics there. This is sort of a fixed way, but you can look at the top 5. You can look at the top 5 nodes like that one, which is the majority of the changes in liver and the associated enrichments. So you can play around with this and just look around. But what's really cool is that there is also an interactive tool. And here you can also compare multiple tissues, or you just select one. Yeah, I was just selecting gastroc. So here you can play around both with visualizations of these trajectories. You can, split them by ohm if you want to see what ohm is driving these changes. You can also download the data. So up here, if you just want to download a figure, you can do that. So there are lots of different ways to play around with this and extract data from whatever you're interested in. So if you go to the cluster composition, you see how many features that follow that trajectory. So here it's upregulation in both males and females across all time points in the skeletal muscle. So if you click there, you then get all of the features that follow this trajectory. So if you're interested in that, everything that was upregulated across all time points in muscle, you can immediately look through that or just download the data and browse it around yourself. So you can see 249 analyzed changed. And there you see those in line plots. And you can also look at the pathway enrichments and see what's driving these different pathway enrichments. So you can see fatty acid metabolism, which skeletal muscle proteins that were changing. You have cristae formation, and you can play around with this however you like. If you want to highlight something particular. So there's a lot of really cool features. So there you see respiratory electron transport. You can see which proteins in the skeletal muscle that were driving these enrichments, which transcripts. So all of these proteins and transcripts follow this time-wise trajectory with being upregulated after both 1, 2, 4, and 8 weeks of training in both males and females. So this really is an amazing resource. Let's see if he wants to move on. Yeah, an amazing resource that is accessible to you. And I think I'm just going to skip this last part because I want to give a lot of time for questions. Of course, we're hoping that this can lead to a lot of cool targeted interventions. So where we can start to get towards more targeted exercise interventions, depending on what the specific health outcomes we're interested in. Like if we want to lower LDL cholesterol or increase our aerobic fitness, or any way we can try and motivate people to exercise, and hopefully be able to be more precise in our recommendations. So tailor exercise recommendations more. We want to be able to enhance performance depending on our baseline and health status, so we can all live longer and healthier and happier lives. And of course, therapeutic development, which I didn't get into there, but obviously a lot of these sort of key molecular drivers of the exercise response are really interesting for as therapeutic targets, especially for treating cardiometabolic disease. So we're starting to get sort of the data to really begin to address some of these challenges. And we can see MotorPack as like it's a baseline study that is really available to anyone. So you can look up and use it for hypothesis generation or whatever you're interested in for your particular research. MotorPack is a huge consortium, and it's really the work of many, and it's very interdisciplinary, so lots of different expertise, and again, funded by the NIH Common Fund. So of course, thank the funding from NIH for really putting in a lot of money into this study. So with that, I want to thank you all for listening, and I'm happy to take questions.
Excellent. Thank you so much, Malene. That was wonderful. Amazing visuals, easy to follow.
Thanks.
Everyone, please feel free to ask questions. We especially encourage students to ask questions, and there's no such thing as a bad question. Okay, so please feel free to raise your hand. And we have a student. Oh no, it's Dr. We have Dr. Hood here. Dave, go right ahead.
Well, I just want to say what a great presentation that was. That was really stunning. You know, I've had the privilege of listening to other colleagues present some of this MotorPack stuff, and your presentation, Malene, was easily the most clear and really educational that I've seen out of 4 or 5 that I've seen before. So congratulations on that, and thank you for that. I really liked it. I just have one really small question, and that's about the UCP1. I thought that it was the males that lost weight or something with training, and yet their UCP1 went down. Am I right on that? One would think that if UCP1 had any kind of thermogenic effect and, you know, helped in body weight control, which I don't know, then it would go up, but that didn't appear to be the case. Any thoughts on that, or did I miss something?
Yeah, no, that's correct. So our hypothesis was with this, yeah, so it went down quite substantially in males, and the males were also the animals that lost fat mass. So our hypothesis was that there was a downregulation of the mitochondrial content in brown adipose, and including UCP1, for the males to sort of preserve energy, really. Because they lost fat mass, they wanted to then preserve energy and not lose too much just by generating heat. But this is, of course, I mean, MotorPack is really a descriptive study, so we need to do more mechanistic follow-up and look specifically. And for the brown adipose was unfortunately one of the tissues where, I mean, it's a small amount, so we were limited by tissue amount, but we only had transcriptome and metabolome, and then we had the epigenomic assays as well. So we didn't have any proteomics data that we could follow up on. So I think that would have been helpful also for interpretation. But yeah, no, it's but you're correct in the down for both UCP1.
Just a minor point. Anyway, thank you and congratulations on this work. It's great work. Thank you.
Thank you very much. I really appreciate that. Thank you.
Other questions? Any students with questions? Or non-students? Okay, I know they're thinking of questions. I've got a question, Malene.
Sure.
what is the time point? When we say there's time points after training, what is that time point in most of these studies, particularly the transcriptomic studies?
So for the most of the data that I presented today, the time point is 48 hours after the last exercise bout. So for the endurance training, they were trained for either 1, 2, 4, or 8 weeks, 5 days a week. And then the sampling was done 48 hours after the last exercise bout to sort of get at the more of the baseline adaptation, so to say, rather than the transient increases or transient changes that you see with one bout of exercise. But we are in the process of analyzing the acute response. So we have from these rats that have done just one bout of exercise. And then we have sampling from immediately after up until 48 hours after. And then I think it's 7 different time points over that time course. So then we'll be able to, it's going to be really interesting. Those data are probably 6 to 8 months from being ready for release. But I think it's going to be really interesting to compare that data with the chronic and see which signals my suspicion is that we'll see pretty different responses between like what is acutely regulated and then compared to this, which is more the long term. But for the humans here that I showed just a little bit of the preliminary results, that's one bout. And 3 time points after exercise.
Yeah, that I saw. That was really interesting. With regards to the influence of the experimental design on the interpretation of results and trying to understand what these transcriptomic responses are reflecting. With a 48-hour time point, 48-hour post training, and I guess that really means 48 hours after the last workout design, as you said, that gives us insight into the baseline, sort of resting baseline. And so, that raises interesting questions. And this goes back, since gene expression, exercise effects on gene expression started as a field in the 80s and so on. But perhaps that design gives us insight into how tissues are maintaining homeostasis after training. In regards to getting insight into the process of adaptation and how the adaptation occurred, those acute studies will give a lot of insight. So when these motor pack studies are occurring, are they sort of discussing the context in which to interpret these results? Like, for example, the post-training doesn't really tell us how it all came to be. It just tells us, it tells us valuable information on, we have a new set point. Is that discussed at all?
Yeah, it is. Absolutely. And I mean, one of the key reasons also for doing this 1, 2, 4, and 8 weeks was to sort of get, we still have these time trajectories that I showed.
Right.
And what's interesting is, I mean, a lot of animal studies out there, animal exercise studies, they are typically 4 weeks long, right? For example. And for the brown adipose, for example, very little happened after 4 weeks. But between 4 and 8 weeks, that's when things started to happen for that particular tissue. And, with the male adrenal gland, we saw this like huge increase after 1 week and then pretty much nothing. So if we would have just sampled, control and then after 8 weeks, we would have completely missed that. So I think you have to select some time points, right? We're definitely more limited in the humans. With regards to time points, because we can't sample, too many times. But with the rats, that's also one of the strengths that we do have more time points. And I'm really excited to see some more of the results from the acute regulation and how we can associate that with the new set points
Mhm.
that we see.
Absolutely. For these rodent studies, any hands up? Oh boy, when there's no hands up. Oh, there we go.
Are
Okay,
there-
Arthur, go right ahead.
All right, I'm Malene. I had a question with the exercise. I mean, I guess it depends on how exhaustive or how tiring the exercise was that will upregulate the genes a bit differently. Like, how did you decide what type of exercise intensity or how long to like run these rats for? And I guess even with the human exercise, the same. Like, how did you decide what type of exercise protocol to have them go through?
Yeah, I mean, that's a great question. And in the humans, we have two different modalities even. So there we have resistance and endurance. For the rats, it's only endurance, and it's the classic like percent of VO2 max is how we determine. And for the rats, they train at the same. But it's it's an approximate per VO2 max, so we don't individualize based on rat. Even though there is some variability between how fit they are. The females and the males train at different. And then it is a ramped protocol. So there is a continuous increase in both grade and speed over the training time course, because obviously they do become more fit. There is also an increase in duration. So I think they end at around 60 minutes, I think is the end. But towards the end, the last few weeks of the training, over these 8 weeks, there was no ramping. So the last few sessions were probably not that hard for the rats. And we do a similar ramped protocol also in the humans.
Okay. And would you expect any of your data to change if you chose to do like HIIT exercise, for example?
I think we would see some differences, absolutely. But yeah, I think with HIIT, we actually have an ongoing study at Stanford where we looking to compare sort of more moderate intensity to high intensity. I mean, there are lots of human studies out there showing that HIIT might be somewhat superior when it comes to increasing VO2 max, for example, over a shorter time frame. So I definitely would expect to see some differences in the molecular adaptation, but I think HIIT will still be way more similar to endurance compared to resistance. Yeah, and we don't have any resistance in the rats, but there are people working on that too, trying developing more good protocols for rat resistance studies
All right.
to be able to look at other tissues. So
Perfect. All right. Thank you.
yeah.
All right, we are right at 4 o'clock. Unless there's any other questions, this has been perfect timing. And well, how about with that, Malene, we all, everybody in the audience, please join me in thanking Dr. Lindholm for this wonderful presentation. I think Dr. Hood put that perfectly. And Malene, we can't thank you enough, and we really look forward to the exciting data from that new study design that you're conducting.
Yeah, more data to come. Yeah, and please check out the MotorPack Data Hub.
Yeah, that tool is very interesting. I'm going to do that.
Check out your
Thank you.
favorite factors.
That's right. All
Awesome.
right. Well, that concludes this presentation. Malene, if you could stay on for a little bit, and everyone else, I hope you have a wonderful weekend. And that concludes the seminar series for this fall. But watch out